Okay, so welcome everyone. Today, our journal presentation presenter is Dr. Ashish Dhar. He graduated from Dhaka Dental College, and after that, he did his master's in public health, and then he finished his PhD in Kagoshima University in medical science. Then he joined in Duke University for a short period of postdoc, and now he is in Mayo Clinic doing his outstanding research in neuroinformatics lab. So welcome, Ashish. Please start your presentation.
Thank you so much. Well, now I'm sharing my screen here for everyone. So is that clear? So now clear everyone, right? Monday?
Yeah, you can slideshow.
Okay. So now slideshow is running on.
Now we are seeing the not the slideshow.
Really? But I am sharing slideshow.
Means it should be full screen.
I did the slideshow.
Now we are seeing your PowerPoint when you shared, but the PowerPoint should be full screen, isn't it?
Now full screen.
No, now you're not sharing.
Okay, just give me a second.
Ashish bhai, I can guide you step by step.
Oh, maybe.
Okay. So select the share screen option.
Okay.
Then select the upper left option.
Share screen.
Then you will see multiple options and select the most upper left one.
Is that clear now?
Yes. There you go.
Okay.
Thank you, Masoom.
Thank you. Thank you for your help, Masoom. So thank you so much. So it is a little bit late for me for the technical support and many things. So this is my PhD topic. So it is very weird that I'm showing the summary, what I'm telling you, but it's very necessary for everyone that what I went through and why I did that. So firstly, I want to say that aging is a problem if we think due to aging, beta accumulation happen in our brain, and due to aging, the most of the cases, amyloid beta accumulation is happen, and we are suffering. Many people are suffering from Alzheimer's diseases, and why the aging happen? So aging is a disease or it's the progression of the lifestyle. So this research actually showing us that uh, aging is not a disease. We have to expect, we have to accept it, but at the same times we have to think about how can we minimize that kind of matter? How can we minimize the uh, penalties we suffered when we are going through the aging process? So here I'm showing that due to aging, amyloid beta accumulation happen, and due to tooth loss, also amyloid beta accumulation happen. And as because amyloid beta accumulation happen in trigeminal mesencephalic nucleus, which is not clear for everyone, but I will show later. It's very much nearby to the Lucas Aurelius region, and why the Lucas Aurelius region is very much important and why we did that. Basically, locus coeruleus is very much important for cognitive function, and at the same time, due to the locus coeruleus region, it's relatively connected with the hippocampus, and hippocampus is very much hot topics for the Alzheimer's disease researcher. So here is the summarized format of my research, and so this is the topics I gonna go through. So my topics was the periodontium damage induces neural cell death in the trigeminal mesencephalic nucleus. So in that time, I was a PhD student in Kagoshima University. Now I work as a research fellow at Mayo. So firstly, I like to share that why and how we became interested about the trigeminal mesencephalic nucleus and what is periodontal ligament and what is periodontium actually. So trigeminal mesencephalic nucleus is basically a primary neuron, and VMS, we called it VMS, and VMS receives the sensation from the periodontal ligament and muscle spindle of the masticatory function. And it is very much important as because mastication is important for not for only biting the food, it also important for speech. It also is important for activation of our neural connection, and it's directly related with the central nervous system as well. So, and the why we think about why we correlate about with the periodontium, why we correlate with the periodontal ligament. If I say that, so in that time actually we thought periodontal ligament actually act as a cushion. It's just like if we bite so hard, tooth cannot touch the socket. If the tooth touch the socket, we would become hard, and it's very it would be very difficult for us for bite or speech. So periodontal ligament also very much important for everybody, and it composed with a fibrous tissue, extracellular matrix, and nerve. It absorbs, as I said, occlusal force to protect the blood vessels and nerve, which is very much pivotal for the tooth structure component. And it prevents the excessive forceful bite. As I say, if we bite so hardly, it would touch the teeth to the socket bone, and if the tooth touch the socket bone, it's really dangerous for the people. So that's why we were very much interested about the trigeminal mesencephalic nucleus and periodontal ligament. So there were many research went out about that method. Before my research, actually, our lab published another paper in 3x trigeminal, 3x TZ mice. So it is triple transgenic mice. So what is triple transgenic AD model mice? So basically, triple transgenic AD model mice is Alzheimer model mice. So these mice actually born with Alzheimer disease, and they have the amyloid beta accumulation from the beginning of their birth, and they have the we observe the phospholipid tau, even though in the same mice. So we examined that mice and we extracted that mice, and we saw and we published a paper about that method. As I showed, tooth loss induces memory impairment in APP mice. So APP mice is different kind of mice. So amyloid precursor protein mice. So that's another topics. But why these topics is the burning issue for everybody nowadays? Because now there are many epidemiological research went out about that, like tooth loss, memory impairment, tooth loss associated with Alzheimer. So the epidemiology shows that if the tooth less, the person suffered the disease more. Person suffered from the cognitive impairment more. And this kind of matter actually make interested to the basic science researcher, and they want to find out the mechanism. So Number 4 is the basement. Actually, there are many research went out about epidemiologically. That's why the Japanese government are very much careful about that. They have the project like Hachimaru Nimaru. So every 80 years old person should have uh, 20 teeth. So they're very conscious about the tooth matter. That's why the epidemiological research uh, make the plot, and after that, the basic researcher showed the mechanism how it induces Alzheimer's disease. So in my research, actually, I was also interested about the bimotor neuron as well, because there is a direct projection from the BMS to the bimotor. Bimotor means trigeminal motor nucleus, and trigeminal motor nucleus is directly uh, related with the masticatory function. And what is masticatory function? There is a there is a normal phenomena that we think like only the bite, only the chewing is the only effect of masticatory function. No, actually, masticatory function means all the apparatus of muscle of mastication when it works, it's called masticatory function. So masticatory function is very much important as because without masticatory function, we cannot talk. Without masticatory function, many neurological disorder happen. So after the BMS neuron, as because it's directed project to the bimotor neuron, trigeminal motor neuron. So we focused on the bimotor as well. And we divided, actually, in my research, we divided the function like we show the amylotropic lateral sclerosis and frontotemporal degeneration and amyloid beta accumulation. This can be happen as well due to tooth extraction. So there is a bargain between the toothless and the tooth extraction. In my here I will show only the tooth extraction cases, and I must want to say that it is happened to the mice case, not for the human case. There is a difference between mice and human every time, but we shows in the wild type mice case, it shows in the genetic modified mice case. So maybe in future people will show in the human case as well. So as I showed here, we divided TDP43, and why TDP43 is important. TDP43 is the RNA translation regulate the RNA translation, alternative spacing, and mRNA stabilization. And there are 3 stages of TDP43, basically cytoplasmic TDP43 and neural cytoplasmic inoculation. Why these two conditions called pathological condition? Because in these conditions, we cannot see the nucleus, even though we can see the crescent shape of nucleus. That's make researcher more curious about the TDP43, because in terms of amylotropic lateral sclerosis, in terms of frontotemporal disease, these phenomena we can observe as well in that kind of research. So here is the research question we set at that time, actually. So after tooth extraction, whether or not venous neuron die due to periodontal damage. As I say, this was our second paper. The first paper went out in the genetically modified mice, triple transgenic mice. This research went out in the BL6 mice, I mean the wild type mice cases. So number 2, we said it about the after venous neural damage, whether or not TDP43 pathology appears in the bimotor neuron. So as I say, bimotor neuron is the masticatory neuron, and it's responsible for the masticatory function. And at the same time, if we see the TDP43 appearance in the motor neuron, there is more or less confirmed that maybe ALS disease can happen in that patient or in that mice. There are many evidence went out about that research. So that is different kind of issues for the ALS researcher. So this was our first paper. Like we extracted the mice, we see the ATF3 positive. When I will show what is ATF3. ATF3 basically we use for the damage marker. And what is damage marker? Actually, when the neuron damage, ATF3 appears. Activatory transcriptor factor 3 appears in that neuron. And after the activatory transcriptor factor 3 appears in the BMS neuron, it also accelerate the macroglial function because inflammation happened that part. And we know if the inflammation happens, microglia become activated. And we were stated the hypothesis about the microglial activation. That microglia actually we thought maybe not helpful for the Alzheimer's diseases. And we showed that after the tooth extraction, after the molar tooth extraction, amyloid beta accumulation increases, even though 2 months cases mice, transgenic mice, and it shows very weirdly cognitive dysfunction. They even ate their own babies because they couldn't remember their babies, even though here I show this last part of the picture. We showed that there is a behavioral test, and this is called Barnes-Maze test. And this Barnes-Maze test actually used for that kind of mice who cannot remember. So in that case, we showed like mice cannot remember the hole where they can escape from that kind of mess. From that paper, we confirmed about the triple transgenic mice behavior and the weirdness or exaggeration of the amyloid beta aggressiveness of that mice, triple transgenic mice. Here is the paper we showed, like after 3 molar extraction, amyloid beta accumulation happened, and that amyloid beta accumulation happened in the cytosol region. And as because BMS and Lucas Reyes region is very much near, so maybe for the BMS destruction, for the BMS activated microglia destruct the Lucas Reyes region as well. So that paper we showed like after the BMS destruction, Lucas Reyes also destructed or also destroyed because of the acceleration or because of the overactivation of microglia. So that was the paper we published at the beginning. Then we decide what happened if we used the wild type mice, because as I said, 3xTZ AD mice, AD model mice, actually the Alzheimer's disease model mice. So they're born with Alzheimer's. So if we use that kind of procedure with the wild type mice case, we can find the same result or not. So that hypothesis went out to my PhD research, and we studied, or in this study, we aim to reveal that the effect of tooth extraction on the neurodegeneration of BMS motor and show the initiation of TDP43 pathology in C57 BL6 J mice. Actually, BL6 J mice as we know, it is a wild type mice. And from this research, we want to clarify the projection from the VMS to the periodontium, and at the same time, how long it takes for the VMS destruction or degeneration. And we want to find out the pathological type of TDP43 in the V motor, because if the V motor damage, if the motor function damage, we can see some effect. We want to see some effect in TD about ALS disease. So these three guideline we followed that time, and at the same time, as because of the wild type case, we want to follow another kind of guideline, because wild type mice case, previously many researchers says that like there is no connection between the VMS to V motor. They showed there is no direct neural connectivity. So that was the challenge for us, that like if there was no connectivity from the VMS to the V motor, how it distracted the V motor. So firstly, we like to show that kind of connectivity in my research. So here is the protocol. I don't want to go through this protocol, what we did. So here is the another protocol we did. Actually, we injected fluorogold and we fixed the mice, and we extracted the molar teeth, basically molar all three, not the incisor, because in the mice case, if somebody have the experience like mice cannot eat without their incisor, so it's very hard to keep them alive if we extracted the incisor of the wild type mice. So we every time we extracted the molar teeth, and we gave them the powder food for their diet. We used this section, and that the procedure we maintain in my whole research. And this kind of primary antibody we use in my research, as we use piezo2 for venous neuron identification, ATF3 for damaged neuron marker. It's important because I will talk about this kind of primary antibody next time. And so piezo2 is for identification of trigeminal mesencephalic nucleus. So it is very much responsible and directly connected with the tooth socket and periodontal ligament. And ATF3 is the damaged neuron marker. If something damage happens, ATF3 appears over their nucleus. And TDP43 pathology actually responsible or appear when the ALS disease happens. And this is the protocol we maintain. This is the primary antibody we used. So as I said, there are many researchers said that there is no connection from the VMS to the V motor. So our goal was that time, like, is there any connectivity present or not? So we injected retrograde pressure in the mice brain, and as well as we injected fluorogold in the periodontal ligament. So retrogradely, we want to see there is a connectivity from the from the VMS to the periodontal ligament, and antrogradely we want to show the same connectivity. So it's a double checkup that time. That is the statistics procedure, and we got the whole result, like characteristics of the VMS neuron projecting to the periodontal ligament, loss of VMS neuron after tooth extraction, neurodegeneration of VMS neuron after tooth extraction, and effect of TDP43 appearance after tooth extraction. This figure actually retrogradely and antrogradely proven like there is a real connectivity from the periodontal ligament to the VMS. And this connectivity we want to show, because if there is no connectivity, so we cannot prove the next step. So this is our first goal. So we injected retrograde and antrograde pressure as I showed here. like 5 days, 10 days, and 15 days case, we found the direct projection retrogradely and antrogradely in the brain, and retrogradely fluorogold into the periodontal ligament. So that part of the figure make us curious that which nucleus, like which mesencephalic nucleus is two types, large type and small types. So which mesencephalic nucleus actually projected to the periodontal ligament? And we found out like periodontal ligament actually projected to the smaller size mesencephalic nucleus. So we showed that smaller size periodontal smaller size periodontal ligament actually projected to the smaller periodontal ligament actually projected to the smaller size mesencephalic nucleus, I mean the bemis neuron, and it after the tooth extraction, smaller size diminished. Smaller size periodontal ligamental connected mesencephalic nucleus vanished due to the damage of the periodontal ligament. So our first goal succeeded, and loss of bemis neuron after tooth extraction. So that is the main goal, that is the main topics of our research. So so here shows the ATF3 positive cell, 5 days, 10 days, and 15 days. And why it is important for 5 days, 10 days, and 15 days? Because after tooth extraction, when ATF3 appears, so we tested after tooth extraction in 1 day, no effect happened. After tooth extraction 3 days, and we got the positive result at 5 days. I mean the damaged neuronal positive marker at the 5 days. So after tooth extraction, neural damage happened within 5 days. So we got, and 5 days cases it rises so high. So immediate after tooth extraction, neural damage happened very much. So it reduces within 10 days and 15 days case. And we don't know why 15 days case it increases. That was our burning issue that time. So this was CAPS3 actually the death cell marker. So if the damage happens as because of the phenomena of the neural cell, like it can be cured. So we were curious about if the damage happens, these neural cells become dead or it become cured. So we tested the CAPS with the CAPS3 marker. CAPS3 is the death cell marker, and we found the positive result that after tooth extraction, neural death happened. And we show that in our picture here, like the upper E and F shows that proven. And in the 7 days case and 12 days case, there is no significance, but there is a correlation. Like the 7 days case, more neural death happen, and 12 days case, after 12 days, the neural death may be gradually down regulated, or CAPS3 down regulated. Maybe the neural death become less in the wild type mice case. As I said, it is a wild type mice. So now about we want to see the TDP43 appearance in the tooth after the tooth extraction. So it was the first time we showed is there any relation between the tooth extraction and ALS present or not. For that purpose, we injected anterograde tracer, AAVGFE, anterograde tracer into the bemis neuron, and we saw there is a real connectivity from the bemis to the B-motor. So it was a success that time actually. So there were many researchers who said that there is no connectivity for the wild type mice case. from the BMS to the B motor, but we showed yes, there is a connectivity from the BMS to the B motor, and this connectivity actually make us curious about the TDP43 that time. So CH80, about the CH80, this is the motor neuron marker as well. So acetylcholine. So we co-staining with the ABVJFP and CH80. So there is a we saw the March picture, and there is a real connectivity from the BMS to the B motor. So there, this was the success that time for my PhD paper. And we, as I said into the introduction, so we got the TDP43 positive cell after tooth extraction into the B motor region. So TDP43, we divided 3 types: internuclear type, cytoplasmic type, and neuronal cytoplasmic type. So these 3 types basically TDP43 present into the nucleus. The nucleus fully damaged for the cytoplasmic type, and neuronal cytoplasmic type. So we can see the crescentic shape of the TDP43 positive. So from the number 3, basically the bimotor neuron nucleus, bimotor nucleus damaged partially. So these 3 basically the alarming issue for the ALS patient. And in that topic, actually, my previous lab, they are doing outstanding research, but I am not part of that research now. So here is the graphical picture. As I said, of course, there is after tooth extraction, TDP43 expression. We can see the TDP43 expression, and it appears. And from that graphical figures, actually, we confirm that due to the tooth extraction, it's responsible for Alzheimer's disease as well as ALS. So it was the beginning of that research. So in conclusion, we prove there was a clear projection from BMS to the periodontal ligament in wild-type mice. That was our success, and periodontal ligament was innervated by smaller size of BMS neuron. And BMS neuron showed the occurrence of cell death between 5 to 10 days after tooth extraction for the wild-type mice case. And as I said, it was another kind of success. Like for the wild-type mice case, there is no invasion, there is no transformation of amyloid beta or projection. So it was my PhD topics was that kind of part. So we got that result, and pathological TDP43 appears. So there is a real connectivity between the tooth extraction and ALS diseases. And that's it. Thank you for that answer. I expect questions. If anybody have the questions about the mice or my research, I'm very willing to answer them.
Yes, I have a question.
Sure.
All right. So we know Alzheimer's is a generalized degenerative disease of the nervous system. That means both hemispheres get affected. My question is, in your research, did you test the contralateral side of tooth extraction? Like, did you test the bilateral side or the one side? And if you test only the one side of tooth extraction, and then which side you pick?
Oh, I am very sorry to say that. I'm very sorry that I didn't say that. We use control like the ipsilateral control, bilateral control. So we tested both sides. We also use the ipsilateral side, like one side extracted, another side not extracted. So we use that as a control part. And at the same point of view, we also tested the pure control. I mean, there is no extraction. So we tested the both sides, and we had the negative control, positive control, and ipsilateral control, bilateral control, both.
Okay, so just curious, is there any difference between dominant hemisphere side versus non-dominant hemisphere side? For example, in clinical practice, when we do the whole brain radiation, if we spare the dominant hippocampus, that preserve memory, that preserve the early onset of cognitive decline.
You are asking you are asking the next research project of me.
Okay, perfect.
No, that was not my business now. So actually, if we extracted, we saw that if we extracted both sides, so bilateral cases, it damages more because because of the excessiveness of the injury, you know. So if we extracted the single side, we thought the massiveness of the extra injury is less. So ipsilateral case, it shows little bit less extraction, little bit.
That makes sense.
Yeah, you are right. But we use both. It's very common.
Impressive. Thank you.
Does anyone have any question?
Hello. First of all, thank you, Ashish bhai, for the wonderful presentation. Interesting topic. So you did the experiment on the mice model, right?
Yeah.
I'm assuming it's not possible to go ahead and ask people to remove their teeth and check their trigeminal neurons. So can we replicate in human, like we can check the trigeminal nucleus of the people who had history of tooth extractions? Maybe can we do that in future?
I'm not sure I'm the right person for asking this question.
To Rafi's question, I have some opinion I can share.
Sure.
Correct. So in human, it's certainly possible to see to look at the trigeminal neuron, trigeminal nucleus if we have the tissue from the after diagnostic autopsy. But I believe there are certain other way to do that to answer your question. Like in human, is it really related to these tooth extraction and onset of the Alzheimer? First of all, our control should be the tooth extraction group and versus non-tooth extraction group. Then if this pathway involves trigeminal nerve, that is the next question. We can also answer this question by the way, there are certain people who undergoes trigeminal nerve ablation for the trigeminal neuralgia. So if we see that pattern, the patient with trigeminal nerve ablation also get affected with Alzheimer. I think by answering these two questions through clinical research may certainly answer your hypothesis.
Of course. I just wanted to say the same things to Rafi, but you answered perfectly. Outstanding. It's happened actually. Okay, so it's true. And Martin answered the whole things.
Yeah, I have also a comment because in the beginning, in the background, you mentioned that there is a epidemiologic study already found that if the patient has tooth extraction, they are more likely have Alzheimer's disease, isn't it?
Yeah.
So the epidemiology already showed that there is a very much correlation with the tooth extraction and then developing Alzheimer's disease.
Yes.
So there is also already is a correlation. So what Ashish did is to find out the mechanism. Is that the real picture that there is is there any means what is the reason why if you'd had tooth extraction, why your the neuron is damaged. So it's an outstanding presentation and outstanding research work you did. I hope you will do further more research like that.
Yeah, thank you so much. Actually, epidemiologically, they showed many ways. Like in US, in Japan, they have the record, like in the health system, so they can correlate very easily. And in Japan case, as I said before, they have the project, they have the national project. They want to keep 20 teeth in the person who are now 80. So it's a big project. So epidemiologically, they did everything. So for the basic researcher, we are finding the way how we can solve the problem.
Yeah, thank you very much. So
Thank you.
what is the take-home message? Brush your teeth.
Brush your teeth. Yeah. Keep the teeth safe.
Yep. Thank you very much. So so now we can conclude our session. And thank you everyone for joining today's presentation. And thank you, Ashish, for a wonderful presentation. And thank you all. Bye for today.
Bye bye.